purpose of quadrant streaking is to get well-isolated colonies. The reason why we want well-isolated colonies is because we want to make sure that we are working with pure cultures of bacteria. So I'm first going to demonstrate the pattern of quadrant streaking on a plain agar plate or a plain petri dish with a Sharpie. So when you pick up your bacteria and put it on an agar plate that you are going to quadrant streak, for your first quadrant you are going to lay down a really thin layer of bacteria. So you kind of want to scribble the bacteria onto the plate to spread it out as much as possible. So for the second quadrant, you'll rotate the plate just a little bit, and then you will use your inoculating loop to go through that original patch of bacteria once, twice, and then you will make about 6 or 7 wide zigzags. Notice that I want to maximize the surface area of the plate so that I spread the bacteria out as far as possible. I really want to utilize all that surface area in trying to dilute the bacteria on the plate. For my third quadrant, I will then again rotate my plate. I want to come in with my inoculating loop at an angle to pull across that bacteria that I already laid down. I'm going to go in a second time and again make 6 or 7 zigzaggy lines. Notice that I am maximizing my use of the surface area of the plate. Now sometimes we do more zigzags in each quadrant and stop after 3 quadrants. But in this case I'm showing you what it would look like if we did 4 quadrants, which depending on your organism and your ability to get single isolated colonies, sometimes you're going to want to do a 4th quadrant. So again, the 4th quadrant is done the same. You pass through the original batch of bacteria twice and then make zigzag lines. Be careful not to touch the other quadrants once you are in your zigzags. And so you should end up with a pattern something like that if you are doing a 4-quadrant plan. If it was a 3-quadrant plan, it would be only 3 quadrants, but the plate would still be full to maximize the use of the surface area. I'm now going to show you how you would quadrant streak on a plate. Now, the first thing you do when you before you start quadrant streaking is you want to make sure you label the plate with your initials and the strain and the date. So you always want to have those 3 things written on your agar plate, and that of course goes on the agar side. Then you're going to light your Bunsen burner. And you're going to be ready to streak your plate. Now remember, the idea of quadrant streaking is we want to dilute the bacteria across the plate. So we need to start with a sterile inoculating loop. So we're going to flame our loop in the flame and get it red hot so that it's sterile. And now that our loop is sterile, it's very hot, we need to cool it before we touch a colony. So we're going to find a plain area of the agar where nothing's growing, and we're going to touch our loop to cool it. And then we use our inoculating loop to pick up a single well-isolated colony. You don't have to pick up the whole colony so much as just touch a well-isolated colony. Now there's bacteria on my loop, and so I'm going to lay down that first quadrant. And I always like to lay down my first quadrant under the area where I have written the information about the plate. And I put that area under my thumb. So I'm going to take and I'm going to use the loop, and I'm going to spread that bacterial colony out on the plate to make a nice thin layer of bacteria. And so this can be just a back and forth sort of scribbling motion. You're just trying to fill in the area as much as possible. Now note the position of my thumb. My thumb helps me keep track of where I've put the bacteria on the plate. So I'm going to hold on to that plate while I flame my loop. It's very important to flame your loop in between each quadrant because we want to kill off the excess bacteria on it so that with each quadrant streak we are diluting the bacteria on the plate and taking fewer and fewer bacteria. Now when I'm ready for my second quadrant, what I like to do is I like to rotate my plate inward about 1/8 of a turn, move my hand, and then pick up the plate. And this way the area that I've already streaked, you can see is right here, is going along here. And so if I start to quadrant streak starting at my thumb, I can see where the bacteria is, and I know I'll be going back through that original area 2 times. So my loop was flamed and it's cool. So now I'm going to go in and I am going to start my zigzaggy lines, going twice through the original area. I'm keeping the angle of my inoculating loop low, and I'm pulling all the way to the edge to maximize the surface area that I cover on the plate. And most of the strains in the lab we handle 3 quadrants is enough. So I've used up about half of the remaining surface area for that second quadrant streak. I place my plate down, I flame my loop. Remember, it's very important to flame it so that we really are diluting the bacteria. And then I again rotate my plate about 1/8 of a turn, pick it up, and then that shows me, I can see that that's where I need to start, and I'm going to cool my loop. And then I'm going to go through that second set of zigzags. I'm going to go through it once, twice, and then I'm going to zigzag my plate and drag the bacteria to maximize the whole surface area. Just be careful that you don't go back into that first quadrant, otherwise you will suddenly undilute your bacteria. So basically what I've done is now I've maximized the surface area and used the whole surface area to spread the bacteria out as much as possible. And from this I should see well isolated colonies so I can study bacteria in pure culture. And always flame your loop when you're done before you set it down.